Hello, and welcome back to the Scientific Talk. Today, we're going to discuss genetic enhancements, smart animals, and the incredible potential of a smart pill that could boost brain power and memory. Imagine a mouse navigating a maze. It moves through the labyrinth, turning left then right, sniffing the air, pausing, and making a choice. For most mice, this task involves trial and error, learning from the memory of past wrong turns. But what if this mouse had an enhanced brain capable of solving the maze faster and remembering its paths longer? This is the story of the smart mouse, a groundbreaking experiment that redefined what we thought was possible in genetic engineering and memory enhancement. The journey began in 1999, when Dr. Joseph Tsien and his team at Princeton University sought to uncover the genetic basis of memory. Their focus was on the NR2B gene, often referred to as the memory gene. NR2B plays a critical role in synaptic plasticity, the ability of neurons to strengthen their connections as they learn and adapt. This gene acts like a gatekeeper in the brain's learning centers, controlling the flow of signals that strengthen memories. Using advanced genetic engineering techniques, the researchers introduced a modified version of NR2B into a group of mice. What emerged was nothing short of extraordinary. These mice demonstrated remarkable improvements in learning and memory. They could navigate mazes faster, remember their paths for longer periods, and even learn complex tasks that confused their unaltered counterparts. But this breakthrough wasn't just about better maze performance. The implications were profound. The experiment revealed that memory and learning, processes long thought to be unchangeable, could be enhanced by tweaking a single gene. The researchers had unlocked a door to understanding how memories are formed and potentially how they could be manipulated. However, as with many scientific advances, the results raise new questions. Some of the genetically enhanced mice exhibited heightened anxiety and timidity. It seemed that their improved memory came with a cost, the ability to vividly remember negative experiences. Imagine remembering every painful or frightening moment in your life with crystal clarity. It could be as much a burden as a gift. Despite these complexities, the research on the smart mouse opened the way for further exploration. Could these findings be applied to humans? Could the NR2B gene be a key to treating memory-related conditions like Alzheimer's or dementia? Or perhaps even enhancing intelligence in healthy individuals? Imagine a fruit fly, just a few millimeters long, navigating its tiny world with instincts refined over millions of years. Now, imagine that this seemingly ordinary fly has a memory so sharp that it can learn in one trial what others would take multiple attempts to grasp. This is not the stuff of science fiction, it's the result of genetic research into memory and learning. Welcome to the fascinating story of smart flies and dumb mice, a tale that reveals the genetic underpinnings of intelligence and the delicate balance of brain function. The journey begins with a critical molecule in the brain called the cyclic AMP response element binding protein. This gene, also called CREB, is a molecular switch, responsible for activating the genes that strengthen neural connections during memory formation. Researchers realized that by manipulating this gene, they could potentially enhance or suppress memory. In one remarkable experiment, scientists added extra CREB activators to fruit flies. The results were astonishing. These genetically modified flies developed what could only be described as photographic memory. They could learn tasks in a single trial, a feat far beyond the capacity of normal flies. For example, when exposed to a smell paired with an unpleasant stimulus, the enhanced flies remembered to avoid that smell with near-perfect recall. It was as if their brains had been supercharged. But there's a catch. With every enhancement comes a potential downside. While the smart flies excelled at learning and memory, their neural systems became overloaded in other areas. Basic survival instincts, such as avoiding predators or finding food efficiently, were diminished. Their newfound intelligence came at the expense of other vital functions, revealing the delicate trade-offs inherent in brain optimization. The flip side of this genetic coin was observed in mice. Researchers created dumb mice by suppressing the same CREB activators. These mice struggled with tasks that their normal counterparts performed with ease. They were slower to navigate mazes and showed little ability to retain information. Watching their diminished capabilities provided stark evidence of CREB's essential role in memory and learning. These experiments extended beyond curiosity. They had profound implications for human cognition. For example, Why does cramming for exams rarely work? The answer lies in CREB. Overloading the brain with information in a short time depletes its supply of CREB activators, limiting memory formation. This discovery has reshaped how scientists understand memory processes, emphasizing the importance of rest and spaced learning to replenish these critical molecules. The work on CREB is associated with potential therapies. Imagine treatments for Alzheimer's disease or other forms of dementia that boost CREB activity, restoring memory in those who've lost it. Conversely, understanding how to suppress overactive memory systems could help individuals with PTSD or severe anxiety, where intrusive memories dominate their lives. As we move closer to unlocking the secrets of genius, we are at the verge of a new era. An era where the boundaries of human potential are no longer set by biology, but by the choices we make with the tools science has given us. What will we choose to remember, and at what cost? Imagine waking up one day and reaching for a pill that could sharpen your focus, boost your memory, and even enhance your intelligence. No more struggling to remember names, no more hours spent cramming for exams. Just pop the pill and let it unlock the full potential of your brain. But can we improve brain function pharmacologically? Scientists have long known about substances that affect cognition. For instance, caffeine is a natural stimulant that enhances focus and alertness. But the vision of a true smart pill goes far beyond your morning cup of coffee. It's about directly enhancing memory, learning, and creativity at a molecular level. One of the most promising candidates in this quest is a drug called MEM1414. Developed in research labs, MEM1414 targets CREB proteins, which play a critical role in memory formation. Think of CREB as the builder of neural connections. The more active it is, the stronger and longer lasting your memories become. In animal studies, MEM1414 accelerated the production of CREB and dramatically enhanced long-term memory in older rats. Tasks that once seemed beyond their ability became manageable, as if the drug had turned back the clock on their cognitive decline. Now, imagine the implications for humans. A smart pill could revolutionize education. Students could absorb complex subjects in half the time. Professionals could master new skills effortlessly, and individuals suffering from age-related memory loss could reclaim their independence. But the idea of a smart pill isn't entirely new. Drugs like modafinil and Ritalin, originally designed to treat narcolepsy and ADHD, are already being used off-label by students and professionals looking for a competitive edge. These substances improve focus and stamina, but they don't enhance memory or intelligence in the way a true smart pill could. MEM1414 and similar compounds aim to go beyond temporary boosts to fundamentally rewire how the brain processes and retains information. The dream of a smart pill is tantalizing, but it also raises critical questions. If such a drug were widely available, who would have access to it? Would it be affordable and equitable, or would it create a new divide between those who can afford enhanced cognition and those who cannot? And what happens to creativity and individuality in a world where everyone's mental performance is chemically optimized? What do you think about it? If you could enhance one aspect of your brain, what would it be? Share your thoughts in the comments below. Don't forget to like, share, and subscribe. See you in the next one.